And we're back. So, G protein with Adenylate Cyclase. Last one we did was G protein with PLC. All right. So, G protein with Adenylate Cyclase starts the same way. This whole, that's a green marker, which probably is more visible to you guys, isn't it? So we're going to do that. All right. So, something binds, comes down. GDP is turned into GTP. The G protein with its nice charged GTP goes over and activates Adenylate Cyclase. Adenylate Cyclase, if you'll notice that it says adenosine monophosphate, so it's going to take AMP. You can remember that that's the next step because that's literally what this name means, is it's making a cyclic molecule out of adenosine. And so AMP, monophosphate, gets turned into cyclic AMP. And so this thing has done its job. Good job. All right, we move on to cyclic AMP talks to pKa. And you can remember A is always with Adenylate Cyclase, every time. Okay? Protein kinase A then takes a molecule, phosphorylates it or dephosphorylates it, and it's a kinase. So it does its kinase deal, and that's it. This is really straightforward. And so you can remember that the smooth muscle one is the one with all the junk, and then the Adenylate Cyclase one is the one without all the junk. So, and this one is used in a lot of things. So I'm not just going to call it the, heart or whatever. So this is a slower transduction method than if you did like axon, if you just did neurons with neuropeptides and then it was a channel. This is a G protein, so remember that's slower. Okay, moving on from this one, let's go ahead and do rhodopsin as well. Okay?
So
yeah, what's up?
It says AMP.
Yeah.
Is that, like, in my notes I wrote ATP. Is that wrong? ATP
I know it ends up as cyclic AMP. I was pretty sure it started as AMP. Okay. It's possible it's ATP and then the energy. Well, the energy is supposed to come from here. Yeah, I'm pretty sure it's AMP.
Okay. Okay. Oh.
So, double check that it goes AMP to cyclic AMP. I'm pretty sure that it does. So, now let's go ahead and do the rhodopsin. Any other questions? Okay. So rhodopsin.
Sorry, real quick. pKa turns into
pKa.
Phosphorylates. Phosphorylates something. Okay.
Yeah, it's just putting phosphates on something. We saw it put phosphates. It puts phosphates onto a lot of things. One of the things we saw it stick a phosphate onto was in the in biochem, in glycogen breakdown, we saw it acting there. So I'm not going to go into detail on that right now, though.
Okay.
So for rhodopsin, let's go ahead and draw kind of this disc in the middle because I like the way that it looked. And then we'll go ahead. So this thing on the left, this is the lipid bilayer, okay? And here's a channel that is letting sodium, it's a sodium channel and it lets sodium go through, okay? And it lets sodium in. All right, so, and we'll get to that in a second. All right, so there are 6 steps. I'm a little bit more organized on this one, so I'm going to give you 6 specific steps that if you know these 6 things, you're golden, okay? First, with a different color marker, I'll do blue so that it stands out. All right, first we have, I guess I'll make it rounded, it's a rhodopsin. All right, first we have this little thing right here, and it is retinal. You might remember this from vitamin A. Retinal is what ends up in the eye. It's the thing that if a photon comes wiggling in and hits it. Then this thing changes conformation, and it pokes rhodopsin right here. So this is retinal, okay? This one is rhodopsin. Rhodopsin is that guy. All right. Photon comes in and hits retinal. That's number 1, okay? Number 2, after that changes conformation, you can remember that rhodopsin, which is embedded into one of the lamellar discs of a rod in the eye. So you have rods and cones. This is inside a rod at the moment. So rhodopsin, and this is a disc, this whole thing. Disc, and it's embedded in the disc. All right. So rhodopsin has been activated by retinal, and that means rhodopsin is going to pull a G protein. All right, so this is why it looks so familiar to the G proteins and why I left that up there. It is a G protein. It has a special name because it was the first one discovered. And so it is called transducin, okay? And this is number 3. Number 1, number 2, number 3. Transducin, okay? All right. And in rods, it's called transducin. In cones, you'd call it cone transducin. Big difference. So whenever I do number 3, I just remember cone transducin is the other one. All right. Moving on, and the 3 I'm going to put on the outside so you don't think that it's part of that. All right. Then the G protein goes and does what G proteins do. It goes and talks to something else. In this case, it is Guanylate Cyclase. So it's a little bit different than the 2 that we looked at already. It's just another thing that G proteins can go and talk to, not quite as commonly. This one is Guanylate Cyclase. Okay, so G cyclase. All right, so it's going to go ahead and take GMP and make it into all right. So this is not guanylyl cyclase. This is what happens when you're on a board. So it actually goes to phosphodiesterase. Yeah, there we go. It goes to phosphodiesterase, which cleaves the phosphodiester bond in cyclic GMP and makes it just GMP. And so you chew up all the cyclic GMP in the cell with phosphodiesterase. Okay? And this is step number 4. Okay? And chewing up all of, so this being turned on is step number 4, and chewing up all the stuff. And then you have step number 5 is over here. This thing is actually activated when there's lots of cyclic GMP. And so cyclic GMP is what's being chewed up. So if it gets chewed up and this is gone, then this closes. And you get no more sodium coming in, and there's no signal transduction to the brain saying, there's light, see stuff. Okay? So, retinal with a photon. Rhodopsin changes, the retinal changes conformation, tells the rhodopsin to go ahead and activate the G protein called transducin. Transducin talks to phosphodiesterase, which then starts chewing up cyclic GMP and making it into just GMP. And then over here we have the sodium channel closing because cyclic GMP is gone and can't keep it open anymore. So you can think of cyclic GMP just kind of like, I'm holding it open. All right. So, and then the last thing is signal transduction to the brain. Okay. And so here's a beautiful brain. And when the sodium channel is closed, you will have transduction to the brain, and that's what goes on. 6 steps. Go ahead and see if you can remember the 6 steps. And hopefully that helps.